Hi, myself Anagha, Assistant Professor, Department of Microbiology, KSR College of Arts and Science for Women, Tirushangoji. Today let's discuss on the topic bioluminescence from the course Environmental Microbiology. Bioluminescence is a property of microorganisms to emit light as a result of series of chemical reaction that occurs in the body. And the majority class of bioluminescence can be seen in bacteria, and let's discuss about the bacterial luminescence in this video. Luminous bacteria are most widely distributed light emitting organism, with the majority of it existing in seawater and the remaining live in terrestrial or fresh environment. Most of the species of the luminescent bacteria are capable of free living, but the majority are found in association with some microorganisms, with some marine organisms like fish, squads, crabs, nematodes, etc. Here in the picture, it is a pinecone fish which utilizes the luminous bacteria and is colonized in the ventral cavity, and this leads to the illumination of the surrounding, and as well as it can be used for the communication purpose. In symbiosis, the bacteria are nourished with readily available food source for the growth, and at the same time the host utilizes the illumination to communicate, to attract the prey, and to protect itself from the predator. This figure is of a deep sea alga fish, and this carries the luminescence bacteria in the light emitting rod, and this will helps in attracting the prey to the front of its mouth. There are some certain obligatory symbionts which are requiring unique nutritional supplements which are exclusively available from the host. Although the presence of these obligatory symbionts have been detected, they are not separated from their host and therefore are unable to be cultured in a laboratory for the further studies. And the light emitting bacteria are classified into three major genera, that is, the most luminous bacteria are classified into three as Proteobacterium, Vibrio, and Photoradus. Species which exist in marine environment are majoritarily categorized into Photobacterium and Vibrio genera, whereas the terrestrial species are classified as the Photorhabdus. And these species within the Photobacterium genus are generally light organ symbionts of the marine animals, whereas the Vibrio species can exist as a free living organisms and as well as symbionts in the sea. Many luminous bacteria are parasitic, with the Photobacterium and Vibrio families infecting the marine Crustacea, and Photorhabdus luminescence infecting the terrestrial insects like caterpillar, with nematodes as the intermediate host for the bacterium. In addition, free-living luminous bacteria that are dispersed in the seawater can often be found in both gut tracts and also in the skin surface of almost all marine animals and non-specific parasites. Here the picture explains that the plant on which the caterpillar rests is infected with the nematodes, and these tiny worms symbiotically carry luminous bacteria in the gut tract. As the caterpillar ingests the plants, both the nematode and as well as the luminous bacteria are ingested as well. So let's see the species. Each species of luminous bacteria will differ in number of properties, including the species growing conditions, that is the nutritional requirements and growth temperature, and also the reaction kinetics of the luciferase involved in the light generation. Luciferase is a kind of enzymes that is involved in the light generation process. However, all luminous bacteria are rod-shaped gram-negative microorganisms with flagella for their motility. Luminous bacteria are also facultative anaerobes, that is, they are capable of growth even under the limited oxygen supply. Despite the physiological diversity among different species of luminous bacteria, or luminance microorganisms can utilize highly homologous biochemical machineries to produce the light. The onset and the energy output of the light producing molecules machinery are tightly regulated under certain central signaling pathways, and by which the induction and repression of the luminous function Lumination functions in the luminescent bacteria are carefully controlled by the cellular factor and intracellular communication. Now let's see in more detail about the reason of the luminescence in the bacteria. Bacterial luminescence is the enzyme that catalyzes the light emission at the heart of the bacterial luminescence. However, the catalytic machinery involved in the continuous light production in the luminous bacteria include not only the bacterial luciferase but also enzymes that supply and regenerate the substrates of the bacterial luciferase. The DNA sequences that code for the proteins in the luminescent system is termed as Lux genes. Okay, and the bacterial luciferase is heterodimer, that is, it is composed of two different polypeptides, that is, two different subunits. First one is alpha and second one is beta. Alpha molecule is of 40 kilodalton and it is encoded by the Lux A, whereas the beta is of 37 kilodalton, that is, the molecular mass is 37 kilodalton and it encodes for the gene Lux B respectively. And this picture here shows the alpha and beta subunits. Now let's see the chemical property or the chemical mechanism involved in the color emissions. The substrate of the bacterial luciferase are reduced flavin mononucleotide, that is FMNH2, molecular oxygen and long chain fatty aldehyde. The excess energy which is liberated from the oxidation of FMNH2 aldehyde and the reduction of the molecular oxygen is released as the blue-green light emission, which can be read by 490 nanometer. That is, the oxidation process of the FMNH2 and fatty aldehyde leads to the formation of FMN and fatty acid, and thus this reaction will lead to a emission of blue-green light which can be read at 490 nanometer, and this mechanism is shown in the picture here. Luciferase function well with a long chain aldehyde of 8 carbon and or longer with tetracanal presumed to be natural aldehyde used in vivo in the luminescent bacteria. The characteristic color indicates the energy level of photon that was produced when the excited electrons on the flavin chromophore returns to the ground state. Thank you. Hope this video is useful to you. Thank you.